Okay, let's unpack this. If you follow the world of drug development, you know the numbers are just, well, they're brutal. The journey from a promising molecule in a lab to a life-saving medicine is long, it's incredibly expensive, and, something like 9 out of 10 drugs that start clinical trials will fail. And that failure so often comes down to one core problem. We're relying on these simplified, traditional models like basic 2D cell cultures or animal studies that just can't predict how a drug will behave inside a complex, living, breathing human body. Today, we're diving into a genuinely fascinating area of pharmaceutical biotechnology that promises to create a much-needed shortcut: tissue engineering. We're exploring how scientists are literally building tiny, functioning human organs and tissues in vitro, so in a dish or on a chip, to dramatically improve how we test drugs and understand diseases. To guide us through this intricate subject, we are honored to have technologist Dr. Rama Yusvana, a senior lecturer from the Faculty of Engineering Technology at Tun Hussein on University of Malaysia. Welcome to the show. So if you look at the big picture of pharmaceutical biotech, what fundamentally shifts when we move away from those traditional models and toward engineered tissues? What's the core difference?
Thank you for having me. I think the fundamental shift is moving from a system of high-volume, low-fidelity screening to a low-volume, high-fidelity system.
Okay, so quality over quantity.
Precisely. We are prioritizing physiological relevance. What really shifts is our expectation of predictive accuracy. Traditional models, frankly, they lack the necessary complexity. The whole point of tissue engineering here is creating these micro-engineered tissue constructs.
Yeah.
You can think of them almost like miniature bioreactors.
Ah.
They don't just have the right cell types. They have the physical structures and the mechanical forces, even the fluid dynamics of a living organ. That realism is the key.
And that immediately gets at the biggest risk factor in drug development, right?
Yeah.
Toxicity. If a model can predict organ failure early, you save years and, well, billions of dollars.
Exactly. And the best example of this right now is probably the liver-on-a-chip system. It's being used for drug testing and safety screening. The liver, after all, is the engine of our metabolism. It processes almost everything we consume.
So these are microfluidic devices, right? This isn't just a tiny piece of tissue sitting in a dish. What is it about the design that makes it mimic a human liver so well?
That's a crucial distinction. It really is the engineering that matters. These systems are microfluidic, which means they use tiny channels to constantly bathe the cells, the hepatocytes, with media. It mimics blood flow.
So it's dynamic, not static.
Right. And that constant controlled flow applies shear stress to the cells, which is critical. On top of that, we incorporate multiple cell types, Kupffer cells, endothelial cells, and arrange them in the correct anatomical architecture. That whole microenvironment allows the liver cells to maintain their function, something they lose incredibly quickly in a standard static Petri dish.
So you're not just asking, does the drug kill the cell? You're asking how the drug is metabolized and whether the byproducts of that metabolism are toxic, because that's often the real problem in human livers.
Exactly. We can actually watch the precise metabolic pathways that hepatocytes are using. It's a process that closely resembles what happens in vivo inside the body. This gives us a really good, very early prediction of a drug's pharmacokinetics and its potential for hepatotoxicity.
Can you kind of quantify that impact? I mean, for our listeners, so what here seems massive, both for the industry and for patient safety.
Oh, cost of failure is astronomical. A single drug failing late in phase 2 or phase 3 clinical trials can cost a company hundreds of millions, sometimes over $1 billion.
Wow.
By using these predictive liver-on-a-chip models, you can flag potential toxicities before the drug even gets to animal trials, let alone human trials. You can compress that preclinical phase, shave years off the timeline, and most importantly, improve the safety of every single candidate that moves forward.
That speed is invaluable. But let me push back on this a little. These chips seem fantastic for acute, short-term toxicity, but can they really mimic long-term chronic exposure, or the kind of systemic failure that happens in a whole integrated body?
That is a critical bottleneck right now. You're right. The chips are extraordinary for short-term studies, maybe up to 2 or 3 weeks, but sustaining a complex tissue for months. That's a huge engineering challenge.
What's the problem? Nutrients.
Largely, yes. Issues of vascularization and long-term nutrient supply in these tiny systems. But the field is moving incredibly fast. We're already seeing multi-organ on a chip systems.
So linking a liver to a kidney, for example.
Exactly. Linking a liver chip to a kidney chip to study how they interact, to look at those systemic effects, it gets us one step closer to mimicking the body as a whole.
Okay, so that's groundbreaking for testing toxicity. But let's move to the next hurdle: understanding the disease itself. Let's say a drug is safe for the liver. We still need to know if it actually works. If you want to attack a tumor, you need a realistic model of a tumor. This is where it gets really interesting for me. How do we use tissue engineering for complex disease modeling?
This is where we pivot to 3D cell cultures. We have
Yeah.
to move beyond those flat 2D dishes, which, they kind of lie to us about how diseases function. 3D cultures give a much more accurate picture of the in vivo microenvironment because they let cells interact spatially, physically, in a way that just happens in living tissue.
And the difference is huge, isn't it? The gap between a cell on a flat piece of plastic and a cell embedded in a real structure. What structurally changes its behavior so much?
It's all about the extracellular matrix, the ECM. In our bodies, cells are surrounded by this complex scaffold of proteins and signaling molecules. When you put a cell in a flat dish, all of those cues are gone. The mechanical stresses, the structural support, it all disappears.
Right.
3D models, whether they're scaffolds or spheroids, they bring back that complexity. For cancer modeling, for instance, we can use 3D cultures to grow specific tumors that replicate the density, the oxygen gradient, and the cell-to-cell connections of a real tumor.
Which is so vital because a huge reason cancer drugs fail is that they can't even get into the tumor. If the drug can't physically reach the cells in the middle of that dense mass, it doesn't matter how potent it is.
Precisely.
Hmm.
These models let us understand tumor growth, invasion into surrounding tissues, which is impossible to study in 2D, and the tumor's response to therapies. We're testing efficacy in a much more clinically relevant landscape.
And what about the most complex organ, the brain? How far can these 3D cultures take us in modeling something like a neurological disorder?
For neurodegenerative disorders, these 3D cultures allow for the recreation of complex neural networks. We can culture neurons, astrocytes, and other glial cells together in a scaffold, and they will form functional synaptic connections. This is absolutely critical for studying diseases like Alzheimer's or Parkinson's. You can actually observe the hallmark pathologies, like plaque formation in Alzheimer's, and see how that affects the network's activity in real time.
A flat culture can show you a cell dying, I guess.
Right. But only a 3D model can show you the systematic breakdown of an entire functional network.
That does raise a question for me about scale, though. The brain is so incredibly intricate. Is a small 3D neural network, by its nature, maybe too simplified? Are we mistaking the existence of a network for true brain function?
That is the perpetual challenge in neuroscience modeling. And yes, of course, they are simplifications. We are not replicating consciousness or, high-level thought. But what we are replicating are the mechanics of the disease at the microenvironment level, the way a specific protein aggregates and starts to damage its neighboring neurons. And by using these models, researchers can test therapeutics in a much more standardized, repeatable setting than, say, animal models. It helps us find novel strategies that target those fundamental disease mechanisms.
Okay, so we've gone from high-fidelity safety testing on a chip to high-fidelity disease mechanics in a 3D culture. So let's shift to the third major application, pinpointing the exact target a drug needs to hit. This is where we get into organoids.
Right, so if 3D cultures model generalized tissue, organoids are a paradigm shift. They actually leverage the biological process of self-assembly to create these miniature, simplified, but functional organs. They're incredibly powerful for identifying drug targets.
Self-assembly. So how do scientists get cells to just spontaneously build something so complex? Where do the cells even come from?
Organoids are typically grown in vitro from pluripotent stem cells, or PSCs. These are cells that can become any cell type in the body.
The master cells.
Master cells.
Yeah.
And by giving them a very specific cocktail of growth factors and scaffolds, we can guide them through the same developmental stages that happen in an embryo until they self-assemble into a structure with all the right cell types and organization.
That is incredible. You're basically growing a mini organ in a dish just by following its genetic programming. Can you give us a concrete example like the intestinal organoids?
The intestinal organoids are a fantastic example. These 3D miniature versions of the human gut, they actually replicate the crucial crypt-villus architecture, all the folds and projections you need for absorption. And they have the full cellular diversity, cells, goblet cells, enterocytes, all of it. This is essential for studying diseases like inflammatory bowel disease, IBD, or colorectal cancer, where the interactions between all those different cell types are central to the disease.
So if a drug company is looking for a target for IBD, how do these organoids speed up that search?
They provide a critical, human-relevant screening platform. Instead of testing thousands of compounds on a generic cell line, you can induce an IBD-like state in the organoid maybe with some inflammatory signals, and then test how different drugs affect the disease features in that miniature gut.
So you can see the mechanism.
Exactly. You can pinpoint the molecular target. We're asking much more specific questions now. Does this compound reduce the abnormal growth we see in cancer cells in this mini tumor? Does it calm down the inflammatory response in the gut lining?
And because you can use patient-derived stem cells, you're getting closer to personalized medicine, aren't you? Testing drugs against a specific person's disease.
Absolutely. The end result is that you can study the disease and test drugs in this hyper-realistic mini organ. And that just expedites the discovery of novel drug targets because you have this incredible visibility into the mechanism of action.
Before we wrap up, we've talked about the power of this, but what's the biggest practical constraint right now? What's holding back the large-scale adoption of organoid technology? Is it cost?
The biggest constraints, I would say, are reproducibility and standardization. Growing these complex self-assembled structures is inherently more variable than just maintaining a simple cell line. Every batch of organoids can have slight variations.
I see.
So for them to become real high-throughput industrial tools, we need robust, automated protocols. We need to ensure that the mini-gut grown in Singapore is functionally identical to the one grown in Boston. The engineering is solving the biology, but the automation still needs to fully catch up.
That is profound. Okay, so what does this all mean for you, the listener, and the future of medicine? We've seen 3 major applications of tissue engineering. First, the liver-on-a-chip systems, giving us accurate drug testing early in development. Second, 3D cultures, which provide realistic disease modeling for things like cancer. And finally, organoids, which are accelerating the discovery of new drug targets for conditions like IBD. Tissue engineering is really transforming drug discovery from this long, high-failure process into something much more targeted, efficient, and scientifically rigorous. It's a huge leap forward. And this brings us to a final provocative thought for you to think about. If these microsystems can be built using stem cells derived directly from you, mimicking your specific biology, your unique tumor, how close are we to a future where every treatment is tested on a miniature, personalized version of you before it ever reaches your actual body? This has been an incredibly insightful deep dive. We must express our deepest gratitude to technologist Dr. Rama Yusufana, senior lecturer from the Faculty of Engineering Technology at Tun Hussein on University of Malaysia, for the enlightenment and his invaluable insights into the topics discussed. Thank you so much for walking us through these fascinating advancements.
It was my pleasure. Thank you for having me.